Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine and best of your health. This is Dr. Sajid Mahmood Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about short question answers, which is relevant to the Advanced Natural Product Chemistry, CAM 5121. You can see over here the short question number 11. Explain the androgens receptor and binding affinity of dihydrotestosterone. Let's see the androgen receptor and its binding affinity of dihydrotestosterone. The androgen receptor is a member of the nuclear receptor superfamily and binds both testosterone and dihydrotestosterone. These receptors reside in the cell cytoplasm bound to heat shock proteins until testosterone or DHT diffuse into the cell and bind the receptor, resulting in dissociations of the heat shock proteins. This enables the ligand androgen receptor complex to enter the nucleus, where it is phosphorylated and dimerized and binds to androgen response that elements in the promoter regions of the androgens regulated genes. After co-activator recurrent, thereby affecting genes transcriptions. DHT has a more potent androgenic effect than testosterone because its binding affinity to the androgen receptor is approximately double that testosterone's, and moreover, it has a dissociation rate about a fifth of that of testosterone. Organ differences in receptor binding of testosterone and dihydrotestosterone results in part from relative differences in intracellular concentrations of these androgens rather than from differences in receptor affinities alone. Indeed, it has been clearly demonstrated that high concentrations of intracellular testosterone can shift AR binding away from dihydrotestosterone by mass action. Moreover, despite there being a single AR physiological differences in testosterone and dihydrotestosterone action are well known and likely reflect variations in AR receptor distributions. Ligand-induced conformational changes to AR that affect stabilizations, local hormone synthesis and metabolism. AR ligand interactions with chromatin, cooperativity of the receptors with other transcription factors, and actions of co-activators and co-repressors. Thus, the local tissues controls of androgen levels in conjunction with numerous other factors drive AR-induced transcriptional responses. And as elucidated later in this lecture, tissues concentrations of androgens, particularly in the prostate, are partly distinct from circulating levels. Like testosterone, circulating dihydrotestosterone is principally bound to sex hormones binding globulin (SHBG) and more weak to albumin. In general, protein-bound dihydrotestosterone is inactive except in some reproductive tissues in which Magalin and endocytic receptor acts as a pathway for cellular uptake of dihydrotestosterone when bound to SHBG. Studies of interactions between a wide array of natural and synthetic androgens and SHBG indicate that the molecular structure of dihydrotestosterone favors tight linkage to the steroid binding site on SHBG. Compared with testosterone, dihydrotestosterone has roughly a five-fold greater binding affinity to SHBG. Binding of circulating dihydrotestosterone to SHBG in highest in young males, that is 0.5 to 2 years of age, 90%, and thereafter declines to about 70% at age 15 and to 40% in young adult men in that age of 18. The increase of SHBG that occurs with aging, that is approximately 1% per year, increases dihydrotestosterone bindings in older men. Dissociation rate constants from SHBG for dihydrotestosterone and testosterone have been measured in human serum and correspond to half times of dissociation of 43 dihydrotestosterone and 12 testosterone seconds, thus further demonstrating the tendency to which dihydrotestosterone binds to SHBG. Accordingly, concentrations of free circulating dihydrotestosterone in gonadal men are very low and would be expected to remain so even when total dihydrotestosterone levels increase in response to TRT. This leads to an important question. Can an increase in circulating levels of SHBG dihydrotestosterone give rise to dihydrotestosterone mediated effects? Let's see the short question number 12. What do you know about metabolism of dihydrotestosterone? Explain history of dihydrotestosterone and who was the first who synthesized this hormone, that is abbreviated as DHT, means dihydrotestosterone. Let's see the metabolism and history of dihydrotestosterone. DHT formed in peripheral tissues is extensively metabolized before its metabolites appear in the circulations. Metabolism of dihydrotestosterone to inactive steroids occur primarily via the initial actions of 3-alpha 17 beta hydroxysteroids dehydrogenase, that is 3-alpha HSD, and 3 beta 17 beta hydroxysteroid dehydrogenase, that is 3 beta HSD, in liver, intestine, skin, and androgen sensitive tissues. When we see the history of DHT, it was observed that dihydrotestosterone was first synthesized by Adolphe Butent and his colleagues in 1935. It was prepared via hydrogenations of testosterone, which had been discovered earlier that year. Dihydrotestosterone was introduced for medical use as an AS in 1953 and was noted to be more potent than testosterone but with reduced androgenicity. It was not elucidated to be an endogenous substance until 1956, when it was shown to be formed from testosterone in rat liver homogenates. In addition, the biological importance of dihydrotestosterone was not realized until the early 1960s, when it was found to be produced by 5-alpha reductase from circulating testosterone in target tissues like the prostate glands and seminal vesicles, and was found to be more potent than testosterone in bioassays. The biological functions of dihydrotestosterone in humans became much more clearly defined upon the discovery and characterizations of 5-alpha reductase type 2 deficiency in 1974. DHT was the last major sex hormone, the others being testosterone, estradiol, and progesterones, to be discovered, and is unique in that it is the only major sex hormone that functions principally as an intracrine and paracrine hormone rather than as an endocrine hormone. In 1968, Brock, Hoske, and Wilson published the first two papers describing dihydrotestosterone. Wilson and his colleagues subsequently demonstrated that 5-alpha reductions of testosterone to dihydrotestosterone was essential for the normal male fetal, genital, and prostatic development. As a result of this seminal work, dihydrotestosterone has been regarded as one of the three major sex steroid hormones in men, joining testosterone and estradiol. Now we will discuss dihydrotestosterone 5-AR 5-alpha reductase. Urologist Charles V. Huygens in 1901 to 1997 first applied the scientific method to the study of androgen-prostate relationships, earning him the Nobel Prize in 1966. The work Huygens performed in the late 1930s and early 1940s, along with his urological colleagues W. W. Scott and C. V. Hodges, has been deemed to the most important body of urological research in history. Now we will discuss the investigator and authors who have discovered and have been performed different kinds of work on dihydrotestosterone in different years. The first investigator is J. Hunter, that belongs to Scotland. In 1786, he discovered the prostate dependence on testes. The second investigator is Breithold in Germany. In 1849, he discovered the blood-borne nature of testosterone, that is first hormone. The next investigator or author is L. Rosica, that belongs to Switzerland, and A. Butent, that belongs to Germany. In 1939, they discovered the isolation synthesis of testosterone and they got a Nobel Prize in 1939. The C. B. Huygens in 1941, that belongs to United States, he published responsiveness of prostate diseases to androgens deprivation therapy and he got a Nobel Prize in 1966. J. Wilson in 1969 discovered the biological role of dihydrotestosterone. J. Ambrotto and McGinley in 1974 discovered the effects of congenital deficiency of 5-AR. The P. C. Walsh, that belongs to United States, in 1975, he discovered and co-discovery of 5-AR syndromes. The E. Stoner and G. Gormley in 1992 discovered the approval of finasteride. The R. Rittmasters C. Rohrborn, that belongs to United States, in 2002 discovered the approval of dutasteroids. Let's see the short question number 13. What do you know about IUPAC name, characteristics and side effects of dihydrotestosterone or hazards of dihydrotestosterone? The IUPAC name of dihydrotestosterone is 5R, 8R, 9S, 10S, 13S, 14S, 17S, 17 hydroxy, 10, 13 dimethyl, 1, 2, 4, 5, 6, 7, 8, 9, 11, 12, 14, 15, 16, 17 tetra, deca, hydrocyclopentane, phenanthreone, and some more: 5 beta dihydrotestosterone, 5 beta DHT, 5 beta androsten, 17 beta ol 3-on, ethiocoline, 17 beta ol 3-on. And molecular formula of dihydrotestosterone is C19H30O2. Let's see the structure of dihydrotestosterone. Dihydrotestosterone consists of total number of carbon atom 17. The carbonyl group is present at carbon 3 position, while hydroxyl group is present at carbon 17 position. The molecular weight of dihydrotestosterone is 290.4 gram per mole. X log P3 is 3.7. Hydrogen bond donor count is 1. Hydrogen bond acceptor count is 2. Rotatable bond count is 0. And exact mass is 290.22458 gram per mole. The monoisotopic mass is 290.22458 gram per mole. Topological polar surface area is 37.3 angstrom. Heavy atom count is 21. Formal charge is 0. Complexity is 459. Isotope atom count 0. Defined atom stereocenter count 7. Undefined atom stereocenter count 0. Defined bond stereocenter count 0. Undefined bond stereocenter count 0. Covalently bonded unit count is 1. And compound is canonicalized, that is yes. The physical state is solid. Cellular location is extracellular and membrane. The side effects and the hazards of dihydrotestosterone is given below. It is harmful if swallowed. Warning: acute toxicity if we take it as orally. Harmful in contact with skin. Warning: acute toxicity if we use it as a dermal dosage form. Harmful if inhaled. Warning: acute toxicity if it is taken as oral dosage form and will take it as inhalation. May cause cancer and it is dangerous as carcinogenicity. May damage fertility or the unborn child.